Okay, then let's get started. So, good afternoon, everyone, and welcome to the Palaeoverse Lecture Series. This lecture series is focused on software development and methodological approaches in paleontology, with the aim of promoting the use of new tools in the field to give researchers a platform to showcase their work. With this series, we hope to shake up the usual type of lecture by having a mixed format, with time dedicated to the presentation, demonstration, and discussion of the software or method presented. Now, typically these sessions will run for about 50 minutes in total, and we will run this series throughout the year, generally on the last Thursday of each month, unless they kind of overlap with holidays. If you're interested in presenting a new method or software you have been developing or implementing, please feel free to reach out. So before moving on, just a quick few introductions. My name is Lewis Jones, and with me today I have Erin Dillon and Will Gertie. The three of us are on the organizational team of Palaeoverse, an organization aiming to bring the paleobiology community together to share resources, reach agreed standards, and improve reproducibility in paleobiological research. And if you want to know more about the Palaeoverse, please check out our website and GitHub organization. I think those links are just being dropped in the chat now. But that's all from me for now. Erin will now give you some helpful tips and instructions for this webinar and introduce this month's speaker. Thank you very much, and please enjoy the lecture. Over to you, Erin.
Awesome. Thanks so much, Lewis. So as Lewis mentioned, the format of this seminar will consist of a talk by our speaker, followed by about 10 to 15 minutes at the end for dedicated Q&A and discussion. So my fellow hosts and I will help field your questions and moderate this discussion. Please note that the seminar will be recorded and it will be made available on the Palaeoverse YouTube channel and on our website. However, we will stop recording prior to the Q&A period in case you'd prefer not to be recorded. So before we get started, I'd like to quickly point out a couple of useful Zoom controls at the bottom of your screen. You'll notice a Q&A button, so you can use this to submit your questions to our speaker, and during the Q&A session, we'll either read out your question or invite you to turn on your microphone in case you'd like to ask the question directly. There's also a raise hand button, so alternatively, you can raise your hand, and we'll call on you and invite you to turn on your microphone. And there's a chat function. So currently you can only chat with the hosts in case of technical difficulties at the moment, but we'll allow you to chat with the other attendees during the Q&A period. And then finally, there's a closed caption button that you can turn on and view the transcript at any time. So lastly, I'd like to highlight a couple of ground rules. Our aim is to cultivate an open, respectful, and inclusive environment during this lecture series where we can all learn from each other. So with this in mind, we ask that you please be respectful and kind when engaging with our speaker and fellow attendees, particularly when asking questions and interacting with others in the chat. Any form of harassment or inappropriate behavior will be grounds for removal from the webinar platform. So for more information, please refer to the Palaeoverse Code of Conduct, which can be found on our website, and I believe it is also being put in the chat now. All right, so I'm going to transition now. I have the great honor of introducing our speaker, Dr. Ondřej Mottl. Dr. Mottl is an assistant professor of plant ecology at Charles University in Prague in the Czech Republic, where he leads the laboratory of quantitative ecology. Ondřej's research interests span quantitative ecology, macroecology, paleoecology, and community ecology, with a particular emphasis on data science. And in particular, his current work focuses on developing quantitative tools for analyzing global biodiversity patterns. So for example, he's developed our rate poll and our package for estimating rates of change using time series of community data, as well as the FOSSILPOL workflow, which we'll hear about today. So without further ado, we're really excited to hear from Ondřej with a talk titled FOSSILPOL, the workflow to process global paleoecological data of fossil pollen for vegetation-based macroecological synthesis. Please join me in welcoming Dr. Mottl to our virtual stage. Take it away.
Thank you very much for an amazing intro. That was really cool. So, as a lot of things have been already said, so this talk is named FOSSILPOL, the workflow to process global palaeoecological data of fossil pollen for vegetation-based macroecological synthesis. So I like to start my presentation with some big numbers. So I like this one where it has been estimated that 7 out of 8 of global Earth system boundaries have already been exceeded nowadays. We also know that more than 3/4 of all ice-free land has been affected by humans. But we still are not exactly sure when this happened. Was it already at the prehistoric time, right? With the ancient people? Was it with the intensive agriculture, or was it during the Industrial Revolution? As has been said, my name is Ondřej Mottl. I'm assistant professor at Charles University and head of Laboratory of Quantitative Ecology. If you want to contact me, I'm now using Blue Sky more than Twitter for political reasons. So I will be talking about fossil pollen and actually how is it very useful as a window to the past of these vegetation patterns, previous ones. If we want to take a look in this, effect of humans and when exactly this happened, this could be one pathway which we'll be using. And for those who are not familiar with fossil pollen records, I include some kind of slow overview of how are we actually getting the data. So normally the process starts that we will find a good deposition environment. It could be a lake, it could be a mire, bog, swamp, but also it could be the bottom of an ocean or archaeological dig. In that place, we will extract a core, which is basically a tube, where here we will see the individual sedimentation layers, here represented by different colors. And in the next step, we will take up samples from these different depths of this core. And some amazing, really skilled people will then look under the microscope and actually count it and identify the individual pollen grains into different pollen types, as we call them. That could be sometimes a species, it could be a genus or a family, depending on the taxa. And since we have relative abundance of these pollen grains, and we also have estimation of the time of the deposit from age-depth modelings, we are able to reconstruct the vegetation history surrounding the deposition environment we drilled. And hopefully this reconstruction can help us tell us about something about the ecosystem in the world. So this artwork has been created by Milan Tyson. I'm butchering the name, but she's an amazing artist. Feel free to check her out. So currently, there is over 6,500 fossil pollen time series, or we call them records, in a Neotoma, which is the biggest palaeoecological database, I would say the most curated ones in the world. So if you want to use this data, if you want to uncover the insights from the paleo data, we really need some tools which will help us to reduce the bias from these dataset compilation. And these bias can come from different places, because we can imagine that different records, individual data cores have been collected by different people, by different equipment, have been analyzed by different people, right? So if we imagine meaning taking several of these core of these records together into single data set is a compilation, there will be quite a lot things which are affecting the compilation which you are creating. It could be the source of the data, meaning where are we actually getting the data? Are we getting it just from Neotoma? Are we actually adding some data from, let's say, other places like Pangea, or even some private data sets which we are putting to the compilation? Additional thing which will heavily influence our compilation is which deposition environment are we selecting. And I already mentioned this in the previous slide. It could be lakes, mires, but it could be also like marine cores, beach cores. It could be archaeological digs. It could be fluvial records, right, etc. And of course, these different deposition environments may have different effects on the taphonomy, right, of the sedimentation rates, etc. So another big part which will be affecting the compilation is chronologies. And this means how the ages of the individual records, how the age has been estimated for record from these steps, right? And there are different methodologies, there are different ways how to do it, and we will talk about a little bit in the detail later on. Of course, big part is also harmonization. We can imagine that if different data sets have been collected and analyzed by different people, these analysts, right, the people who are actually counting the pollen can have different scales, can have different reference collection, can have just be different interest what they're doing, meaning, and this will change the harmonization precision of the data set. So we need to do some kind of harmonization to put everything within the compilation to the same level, I would say. And of course, at the end, it's also important to have some kind of filtering, some kind of quality check to make sure that all the data we want to be there is actually there and nothing else. So preferably, we would like to do this at macro scale, right? This is what I'm interested in. And I think this is the big benefit of now of this having the data publicly available in Neotoma is that we can do this macro scale analysis. We also want to have the dataset compilation to be project specific, meaning your research question or a project can have very specialized, specificity, what you are interested in, and that could drive the way you are preparing your this compilation. And of course, we want to have control over the data quality, which we are having, and which I think is very, very important, to have reproducibility of how this compilation have been created. And this is kind of very often omitted in lots of the studies, where the process of actually preparing, filtering, and selecting and changing the individual dataset to the compilation is very rarely shared reproducibly. So luckily for you, I have been a part of a project called Human on the Planet Earth in Bergen, and we together created a project called FOSSILPOL, which is the workflow to process such palaeoecological data of fossil pollen into vegetation-based macroecological synthesis. And the reasoning behind, or the reason why we did this, is we, in the team, we were actually doing that. We were trying to do some kind of macroecological synthesis of fossil pollen records around the world, global ones. And once we started preparing the data, we found out that there's actually quite a lot of steps, and it's not trivial to do all of these. And in the process, we decided it will be actually kind of useful to create it into a tool which we can share with the community, so other people can also use this and follow this. So in order to showcase the FOSSILPOL, I decided I will have example project, right? And we can imagine that we would like to know what were the patterns of past vegetation changes in Scandinavia for the last 8,000 years, right? That's kind of a research question. And I selected Scandinavia because this project, when we were creating it, I was actually based in University of Bergen in Norway. So this is it. This is the workflow. This is all the individual steps you have to take. And I imagine this is a lot. So let's break it down very slowly, step by step. Okay, we are going from the top left to the bottom right, and we start what is actually going in. What are the inputs of the FOSSILPOL workflow? There's quite a lot of them, and we don't have time to go each of individual ones, so I will highlight just the ones which I think is the most important one, and that's the config file or configuration file. So it's kind of user-defined criteria guided by the research question, which will tell us how the workflow should behave, right? It's controlling its behavior and all the processes. And it's a file, therefore it can be shared, right? So this is increasing the reproducibility. We're kind of saving all the individual configuration. We kind of select at the beginning of the process to make sure we can then share it with other people, which will be then able to replicate the same compilation as we created. So this goes in. The first step at this big world is sourcing. Where are we getting the data, right? So the main source already has, as I mentioned, in the FOSSILPOL is Neotoma Palaeoecological Database, and it's the default one. Of course, there is possibility within the workflow to include other data, for example, private ones, which some cannot or don't want to or even cannot upload to Neotoma. There is still a way how you can do it, and there is specific pathway to do so in the documentation. There's also first steps where we actually select the data. So first things first, we can select the spatial extent of the data. Which part of the world are we actually interested in? Is it global? Is it just continental, etc.? And much more. Again, sadly, we don't have time to go into much of the details. So in this example, in the Scandinavia example, I created this rectangle, which is the extent, the blue rectangle you can see here. It's the extent of the data which I am interested in this project, let's say. And you can see the individual blue points are all data sets returned from Neotoma. These are all the pollen data set in this rectangle. So and there are some additional steps like spatial elimination, selecting of ecological group, deposition environments. Again, we don't have time, sadly, to go over them. But the next big step is chronologies. And as I said, this is the estimation of the position of each individual samples. And we highly, highly recommend that we re-estimate this again. We will redo all age-shape model again using the newest probabilistic methodology. And this is because lots of the data in the compilation, there could be different datasets which use different methodology or have different criteria how this was created. We want to make sure that all of them are on the same level as the newest one. So this will be done automatically within the workflow. The workflow will automatically add newest calibration curve. This is needed if you use radiocarbon dates for the dating that needs to be calibrated. The curves are automatically assigned by geography of the core. And if needed, automatically the post-bomb calibration curve will also be used. And the main thing which is actually happening there is that there are something called chronology control tables, which is actually the table having all this information about the radiocarbon dates and other types of data used for this re-estimation. And as a user, you can kind of select which, for example, of the types should be used. Should we just be using radiocarbon dates? Should we be using biostratigraphic layers? Should we be using, uranium, tephra, etc.? There are lots of types, and it's up to you to select which of these types you want to include in re-estimation of this, is rerunning these HM models. And of course, you can also select that you will only include cores if they have good chronology, whatever you, and this is, we measure it as number of dates. You can imagine the more radiocarbon dates you have, the better your HM model will be. And of course, you can just be okay with 2, you can increase it to 5, and the more you increase it, of course, the potentially better HM model you have, but of course, the number of records or cores which are fulfilling this criteria will be decreasing stably. And this is a big topic, right? I think this is one of the biggest part of the FOSSILPOL is actually this whole process of re-estimation. Again, this was just a taster. In the example I was showing the Scandinavia, I selected some of the criteria of the control point types, meaning I include only radiocarbon dates, and I selected, I think that it has to be at least 5 radiocarbon dates in each core. And as you can see, the purple ones are those which fulfill my criteria of what is good, solid HM model, and all of the gray ones are those which didn't. They fell out. So the next step is actually harmonization, which we discussed a little bit. And the aim of this is to reduce the effect of taxonomic uncertainty. And it's using, as I described, harmonization tables. So we can imagine that different people name different taxa, may name them differently. It could be sometimes just pizza, it could be, pizza type, it could be pizza ABS, right? And we kind of aware that these are all potentially the same taxa. It's just the different palynologists name them slightly differently. So we want to use a table which will use these different names and put them to the same harmonization level. And actually about this process, we have published a paper, which is Approaches to the Pollen Taxonomic Harmonization in Quadrant Palynology, with the first author, John Burks, which I believe is also here in this call, hopefully. And you can go ahead and read all about this in detail. I would say a bonus in this, if you go to this paper, in the supplementary materials, we actually published continental harmonization tables for all continents except for Africa, which can be used as kind of a basis for building your own harmonization table for your own project. It's kind of, baseline. So, and the next and the last big step is filtering. As I said, we can filter and select samples and whole data set based on different criteria. And the aim is to increase the data quality for my research. It's completely optional. You don't have to do any of this, but I highly recommend looking into them. It could be a filtering based on pollen sum, meaning we will only include samples which has certain amount of pollen grains counted. It could be age limit, meaning we only include whole data sets which span certain age, right? We are interested, let's say, 8,000 years. So they should span at least 6,000 years in between these. It could be age extrapolation, meaning how far we allow the age model to extrapolate the samples. What is, it's kind of playing with the error of the extrapolation. And of course, the minimum number of samples. We only include data set if they have, let's say, 5 samples in the period we are interested in. And much, much, much more. Again, I use some of my set criteria for our example in Scandinavia. And again, I would say the red pink one are those which fulfill this criteria. So as you can see through this process of using the FOSSILPOL step by step, we are kind of started with 300 records, which are neotoma bounded by geolocation, going through to only 100 records at the end, right? Which seems like we are losing quite a lot of data. But my argument would be that we are actually increasing the quality of the data for the specific question we posed at the beginning, meaning what were the vegetation patterns or vegetation, changes in the vegetation in this 8,000 years in Scandinavia. So, and of course, at the end, there are some outputs which are which is pollen diagram. So automatically you will get all pollen diagrams for each individual record printed for you in a PDF file, the HDF models themselves as file, graphical visualization in this kind of overview of the data you have, the dataset compilation, and citation metadata, which I will talk a little bit more in detail now. So again, the dataset compilation is the main output, which is actually RDS file in R, which you can directly load and use for analysis. There's kind of graphical visualization, which is spatial temporal overview. This is for the example we just used. So it'll show you where exactly it is plotted in the map. And also what's the temporal overview, right? Here, every line is actually one of these 100 cores in this map, and we can see what the temporal distribution is. And of course, it's between 0 and 8,000 years before present, because this is the period we set it up the workflow to work, because this is the period we were interested in. And they're just colored by WWF biomes for visualization purposes. And also, which is something which I'm very proud of, there is also automatically outputted citation metadata, meaning that for each of the data, for the whole dataset compilation, you will be also output the name of the owners of this individual dataset with their emails if they are present in Neotoma, but also DOIs of all individual records there and you can contact them for quarters or at least acknowledge them. All of this, including the citation metadata, including the config file, including shapefiles and any other things, are then also automatically zipped into what I call reproducibility bundle, which is a file which you can share with your paper if you're using this compilation for some research question, and you can share it, and anyone potentially can then use this reproducibility bundle to replicate directly exactly your data and get to the same point. So to sum things up, FOSSILPOL is a workflow which handles all the steps and flag places where a user needs to specify the choices. It's kind of talking to you a little bit in R, and I will tell you exactly how this is working in the practical session. And alongside it, we also published a paper called A Guide to the Processing and Standardization of Global Palaeoecological Data for Large-Scale Synthesis Using Fossil Pollen, very short name, of course, which was published in 2023 in Global Ecology and Biogeography. And I was very, very, very happy and excited that actually I found out very recently that we've been awarded as top cited article for that year. So that is a good sign for me that the community is actually accepting the suggestions which we lied in the guideline, and they're using this, which I'm very, very happy to hear. Of course, with project sites like this, we also have a website, which has all the general information, all the links you need, and extensive documentation, which I'm very happy about, because this is a complicated thing, right? So let's jump hopefully a little bit to show you some of the websites. So this is the website of FOSSILPOL project, where we have some kind of summary. We have different sections of the project, the mentioned paper, which I just said, the FOSSILPOL workflow itself, but this website, but also the Scandinavian example, which I just showed. You can also access it and see the individual data and how each of the steps works and what is actually on this website. And I will kind of shortly show you the website, which I think is important. We have kind of general information about the workflow with a figure of the workflow itself, and two main topics: how to obtain the workflow and how to set it up. And I will show you this exactly in practice in the practical session. And I think the main part of this website is step-by-step guide, which is actually explaining more in detail. And you can see the workflow looks more difficult here, right? More detailed here, more in detail how each individual step-by-step is working from. And here we see the individual section, sourcing, chronologies, harmonization. We can jump to harmonization or chronologies, and you can read all the individual nitty-gritty, including the description of individual R scripts, which are present in the workflow, and hopefully are well documented. So let me jump back up. There's also an important part to get in touch, right? If you want to contact me or other other members of the team, or if you want to report it doesn't work, right? It's a software, of course, there will be bugs. So please report them, follow this instruction how to do it. Can we add this? We also kind of added a feature request, and we have kind of a list of potential things we want to include in the next iteration of the software. There's also resources where there is the publication itself. There is any presentation, if you can, any of, the one which I'm currently giving is not there yet. I will add it after the recording is available. There's also posters, etc. Oh, no, I'm jumping back. So speaking of the presentation, for those who are watching this on YouTube, you can just take your phone. There's a QR link which will get you directly to this presentation. You can click all these links and buttons as you want, but I will also make it publicly available at the website. And I believe this is everything from the theoretical section. I went through kind of fast, but the time is pressing and I want to really show it in practice in R. So let me jump to RStudio. Hopefully you can see this. So, and this is the project of the Scandinavian example, which is publicly available on GitHub. And I will kind of show you some small pieces there which you can use. So I'm using RStudio. I normally don't use RStudio. I try to set it up as default as possible because this is the most commonly used IDE for people who are doing this. So there is some kind of README file which you can look into this. But what I will show you, oh no, I forget. Important thing, I forget how to get the workflow. That was very important. So I'm panicking now. No. So let's jump back to the website. If you look into general information and we go how to obtain the workflow, there are just two ways how to get it. Either if you have GitHub accounts, you can use it as GitHub template and just use the template to create your own repo. If not, you can simply click this release page, hopefully, and you should be able to download here the source code as zip file. If you click it, it'll download the whole project, which you can then open in RStudio. So imagine we've done that. And we are now in RStudio, actually within the project. And first things first, we need to initialize the project. So here I'll be using, hopefully you can see, this is kind of the explorer of the individual files. And all the R files are in surprising R. And the first script which we need to run is init. This kind of, as it sums up, you need to initiate the whole workflow to set it up on your machine. And as I said, reproducibility is an important part of my work. I'm trying to make sure that everything works everywhere. So, and you will just simply follow the steps, step 1, step 2, synchronize packages of, as we know, dependency issue with the project, everything. We are using RN. If you're not aware about this, you can read about it online and just loading and installing lots of packages to make sure that this is actually working. The next big important script which you look is the config file, already mentioned one, where here we kind of have a description what it's there, and we can see it's a configuration script which variables that consist through the whole repo, through the whole project. It loads packages, define important variables, etc. And we most importantly, we use this user flag in the script because it's a lot of code. And these are really the variables that you need to be focusing, you as a user should be focusing on. So if I scroll a little bit down, here is the first user, right? So which means user, pay attention, there is something you should set up. So for example, here we define the directory for storing big data files because there is lots of processing happening during the workflow and lots of data being saved somewhere. We give the option to the user to choose where it's saved. By default, this is within this project, this folders, right? But you can imagine you can save it to OneDrive or Google Drive or some shared documentation, if a shared drive, if you want to work different people with it, etc. But this is first thing. Other thing, let's example. Here we have geographical boundaries, the spatial extent, as we discussed, longitude min, max, alt, and this is the rectangle which we set up for the Scandinavia, for example. And you can change it as you want, right? We can do 10 save, right? Depending. And then we can then share this file and it will have all this information with it as the configuration. And there's quite a lot of them. Let's jump, for example, to the filtering section. And here, as I discussed, these are the filtering criteria we kind of set up. You can filter by pollen sum, minimum number of gains 100, target number of gains 150. We can also filter by number of levels here in this row. And we actually set up here what is the minimum number of levels for our compilation. And many more. And in the end, I actually set up also some graphical option to make sure that all the graphics all the outputs of the visualization are same for the whole project. So this kind of general structure of what I'm doing. And then if we look again we want to actually run the project, right? We look here in the Explorer and we kind of number things. So there's data processing, which is the FOSSILPOL repo. Then there is main analysis. And we kind of envision that you will be using this FOSSILPOL not only as the getting and processing the data, but also for doing your own analysis. It could be diversity estimation and exploring rate of change using the rate poll, which has been mentioned, right? So we imagine you will then add more of your script in this main analysis folder, but the majority or all of the scripts needed for the FOSSILPOL are automatically created in this data processing. And these are actually the individual section we see previously, right, in the presentation: sourcing of the data, chronologies, harmonization, filtering, and making the outputs. And we will be specifically in this run. I want you to show you the and if you want to just run the whole thing, if you want to run and prepare all the data, you select your configuration file, just run it. There is a master run script, right? And basically it's a script which is calling other scripts and it's calling everything. So if you source this, it will do everything for you. I'm not going to do it because I think the cool thing about the workflow is that you can go deep and you can go into individual section and run it script by script if you do prefer, or you can run the whole thing, right? So for example, here in the Neotoma source, you have individual steps or individual scripts which are actually being called after each other. You can either, again, call this run11, which is, again, a script which will call all the additional scripts in this one, or you can actually one by one call the script for the outputs. And this is exactly what we're going to do now. So I will not do Neotoma download because they will take a lot of time. So I will show you the extract samples, right? So each script has a header, has a name, what it's actually doing, and there's short explanation for what the script is about to do. And there's the individual codes split into the section, which you can click. Of course, if I'm interested and I'm not sure what some of these things means, because, for example, I'm not that good at coding, I can jump back into the website real quickly, hopefully, and go to step-by-step guide, and I can read the documentation. We go data processing, we go Neotoma source here, and here is the exact extract samples, which is the script I'm looking right now in the RStudio. I was showing it, and here you kind of try to describe what is going to happen. So if you read this, we are actually going to extract the unique dataset ID and their meta information, and also extract the authors of the individual data and save them, right? This is what is going to happen with using this script. So if you go back to the RStudio, and this is the script extract samples we just read about, and I can just source it. Hopefully it will work. Yeah. Okay. So there's lots of things happening. And I will try to explain step by step. So I build it in a way that the R is kind of talking to you. The workflow is telling you what is going on. So we kind of source the script. We said run everything within the script. You can run it line by line if you want. I run it as the whole script. So it says, right now I have main header which is saying I'm processing Neotoma files. So the downloaded files, I'm loading the file. It will automatically load. It will automatically load the Neotoma from 2023. The June 19, 2023, which is I at the time when I was creating this example from the Scandinavia. Successfully loaded. Now extracting Neotoma meta information. It checked that everything is prepared. It counted that it was 289 records at this stage of the workflow, right? It extracted some meta information. It extracted the individual samples, and it checked that both meta and sample are extracted for all of the records. So there are lots of internal checks happening automatically for you. And then it actually checked that it's extracted 76 authors of the data, right? So this is the meta information I was kind of talking about. And then it saved them into this small database, let's call it the database, about the project, which currently have this 288 data sets of 76 authors, and have not found previous version of the file, saving a new file. So it then saved the outputs. So this was very simple, right? We didn't have to do anything because the config file, if there was anything we needed to do, it's already automatically happened. So this is just an example how things looks if you run the workflow step by step. Let's continue, right? Let's continue with Neotoma at this beginning. I'm just checking time. I'm pretty good. Okay. The next script is filter depositional environments, right? That will be the next script in the line. And what is actually happening, as the name said, we are filtering the records based on their deposition environments. And there could be pond, natural lake, etc., right? But because the way the neotoma is structured, it's actually hierarchical layers, and I will show how exactly this works. And this will be an example of first of stop check, something we call stop check. So at this process, the workflow will stop and let the user check something, let him, her or him decide something, and then continue, right? How this works in practice, I can go back to R. I will go into filter deposition environment. So again, we can read that this is filtering based on deposition environments. It will get the deposition environment and filter them by selected types. If I press source, if I run the whole script, let's see what happened. There's a lot of text happening. I'll try to explain. Again, it will automatically say that it's doing it, that it loaded the file we just created, right? Which is today's date, which is now, which is the previous step. It automatically loaded the previous step. Starting preparation of deposition environment selection. It gets a list of all deposition environments from neotoma, but it says that there is an error, right? Selected file not present. There is something wrong. And it will automatically stop. As you can see, the script stopped. It didn't continue. And it says, please open file, name of the file, in exactly where it's stored. And then select which deposition environment should be kept. Include should be true or false. Then proceed and rerun the script. Something you don't see as a user is, in my computer, the Windows, if you're running on Windows or any other operating system, it will automatically open the folder with the file you should be editing, right? And as you can see, this is the deposition environment selection with today's date. I will open it. This is the stop check. It will stop. I need to check something. And the workflow, what actually happened under the hood, I will make this bigger so you can all actually see what is happening. Under the hood, what happened, the workflow checked my 280 cores and look inside which deposition environments are present in my compilation at that time and output just those and show me those. And I need to now select which of those do I want to continue having in my compilation and one which should be filtered out. So explain how this works. So the first column is actually deposition environments, and neotoma, they're kind of stored in hierarchical level. So bog is inside of mire, which is inside of palustrine. But there is not higher level than palustrine. So palustrine will just repeat because there's the highest level, right? You can do the same with fen, mire, palustrine, etc. And here the important part is the include. And so it says in the R Studio, we should decide if we do want to include this, yes or no. And we can simply switch this to true. And we wanna box, I don't want a coastal, so I will keep false. I want a lake, I want a fen, I don't want again marine ones. I want a red lakes. So this is for my project specific. It's up to you to select which do you want. And I will take all of this and I will not select terrestrial and I will not select unknown. Right? So now I did the selection, I went one by one and I selected which of those I want. I save the file, I close the file and I go back to R. Include equals true or false, then proceed and rerun the script. So let's do this. Let's rerun the script. And now something else is happening, right? We can see new text. So it's actually said the list of all deposition environments was loaded, and there has been a selection of 16 deposition environments, which is the selection we made in the file, and actually listed all of them which we want. And then it says number of records prepared 275. So we see like we've lost 13 potential records in this one based on the selection. And again, it says have not found previous version of the file, saving with the RDS format. This is happening because the workflow is slowly saving the in-between steps of the workflow, so you can go back to them as you want. If we run the script again, run the script again, then at the end, it actually found older file, did not detect any changes, not overwriting. And this, I built a system where it will actually detect if the file which is trying to save is different from the previous one, and it will not save it. It will only create a new file or create a new file if there is any significant changes. This is kind of similar for those users who know targets, right? I created this before I knew about targets, so this is kind of me reinventing the wheel. Okay, so these were kind of just a showcase of two scripts or very small portion of the whole workflow. As we see, again, I will show you these are neotoma sourcing, merging and geographic chronologies, harmonization, all of this, and output, which is creating the outputs. And you can run the whole script by just running the master. It will all do all of this for you, or you can run each individual scripts. Of course, there are R scripts. So if you are skilled, R magician, I would say, you can continue and edit these scripts, add additional steps for you if you want, right? This is your project now. It's up to you to do whatever you want. But we created mostly using these different functions from a package we created for you, right? So if you don't know, if you can't code that value of aware, you can just run them and they will do hopefully their magic. Let's jump into something else and show you a little bit more about the output which we are getting. Of course, as we see, we are getting the data assembly, right? So there's the RES file, which actually has the individual records. I will not go into deeper, but it will automatically also output some cool figures, right? It will output pollen diagrams, and we can open, can we? Okay, so, automatically it will create using a Ryoka package. I think I'm pronouncing it hopefully right. I think so. So it will create the pollen diagram for if you want to observe each of them individually. They are there, they automatically output, right? And all of them in the compilation are there. We can also look into some different outputs, which will be chronology, right? We can also export for the same data set or different data set. We can take a look at the age model, right? For those who know a little bit about age modeling, this is the age model for dataset ID 317, which is in the compilation. And we can look if the age model is good or bad. If it's bad, we can just toss the record out, right? We can do some visual check about the data also. What else do we have here? I want to now jump into the meta and references. I think I need to open this in a file. Let's open. I need to close this. Sorry. All right. And again, this is the project. Oh, sorry, my computer is dying. I'll make it bigger so you can see what I'm actually doing. And there are the outputs. There's the meta and references, which I think is pretty cool. And specifically, there is this either data assembly meta, which I would also want to show you, which will automatically output, and that's kind of a summary about the dataset compilation which we created, right? And here you can see the individual dataset IDs. Here you can see some additional information like data handle, ID, site name, longitude, latitude, altitude, like meta information, right? Country, etc. But some also information about like number of sample, what was the smallest and the largest age of the sample, some information about the specification of the age model or age which has been done, and DOI of this record, right? And I added WWF biome for the visualization. As you can see, this is not there automatically. This is something I added. But what is important, there is also author table, which I'm very happy about. And I kind of mentioned this already in the outputs. And that's showing you all the authors of the data present in the dataset compilation. And we can see it in John Burke's. So these are the names, emails, if you want to contact them, which are in Neotoma, but also, and these are the individual dataset IDs. They are actually, they uploaded to Neotoma, right? So we can see here in Scandinavia, of course, John Burke's is very proliferate and done a lot of important work there. I am taking 40 minutes. So I suggest that we go into the Q&A. If this this works for everyone.
Great, thank you so much.
Erin?